So that's the point. We like bevacizumab, at least in some patients.
Well, hazard ratio is 0.72. So then
know.
you come in with these hazard ratios of 0.48, for example, and Aurelia very close to that. Again, where do you get the biggest bang? Now, obviously, you're buying more months with that 28% improvement up front, but that's sort of the question.
But that's the point.
Yeah.
Let's use both. Let's use PARP and Bev.
Well, and Bev after Bev, we do have data.
We can do that. So
right?
we can always, at least in the U.S., we can always use Bev again.
Yep.
So that's the third trial at ESMO.
Right.
Bev alone, and it was a European trial because when it was done, we didn't have bevacizumab approved in this country. But still, it's a global sort of approach. Bevacizumab versus olaparib layered in all comers in the maintenance phase. Right. What do you think of that result?
Well, PAOLA 1 is the trial you're talking about, and again, very impressive data. They allowed, neoadjuvant and so forth. They also had a 2-to-1 randomization similar to PRIMA. They gave the maintenance Bev for 15 months, and then it was randomized to either olaparib or placebo for 24 months. So that was the design of that trial. Large trial again, 762 patients enrolled in the 2 arms. And again, I think it provides a great option for those patients who start on Bev, right? They're having a benefit, and you don't know if you want to take that benefit away. And so this provides the option then at maintenance to add a PARP inhibitor. Now, there's some things you need to think about with this trial, and one is toxicity. Cost, we can discuss if we have time, but real toxicity to the patient, it was higher. Dose reductions, dose interruptions, and discontinuations
for
were
Bev
higher.
was 6% versus 20%.
That's right.
And you can see that we're getting better at Bev. When we did our phase 3 trial in GOG 218, it was 21%. Now it's 6%. We're better at managing the hypertension. We're better at keeping patients on treatment. And if you keep patients on treatment, they have a higher potential to benefit. But with the combination, you're right, it's more toxic. And we haven't talked a lot about toxicity. There's a small, maybe 1 to 1.5% of leukemia. And you got the GI toxicity, the fatigue, and the bone marrow toxicity. Right. So I still like it, though.
Well, I like it because it gives you that option for my 40–50% of patients I have on Bev. In your case, it's going to be higher than that, perhaps. But and then you feel confident that it hit the mark, right? The only thing is, Bev only arm. So all these trials are sort of missing one arm, especially Vilia and PAOLA one, in my opinion. The contribution effect there of trying to separate out Bev versus olaparib in that arm, but the hazard ratio was 0.31 for those that had a mutation, and then I think overall it was 0.59.
Let's talk about 0.31. So people say, well, that's as good as Solo 1. So why don't I just use olaparib alone? Let me tell you, you teach me all this stuff. So if you can't beat something, compete against nothing. Yeah. Well, this is the only trial that actually tried to compete against something, and it still won.
Yeah.
So the confidence here in the therapeutic benefit is even higher. So even though the hazard ratio is virtually identical to olaparib alone, olaparib was versus placebo. PAOLA 1 is versus bevacizumab. And there are a lot of patients in the olaparib alone BRCA mutated cohort that are not cured. So you can improve on that with the combination. So I used to kind of stop the bevacizumab when it was a BRCA mutated patient and pivot to olaparib. Now I want to get both.
Yeah. No, I understand.
And why? Because I'm hoping that I can cure more patients.
Right. No, I completely understand the rationale. Not allowed to look at the medians because for so many reasons we just looked at them. But by far of the 3 trials,
they're the biggest.
And to your point, active comparison versus placebo in the other 2 trials. So it is something to think about. Right. For me, in practice, I haven't completely synthesized all this data. However, I think for me, for those that are already on Bev and I'm seeing benefit and they're not having too much toxicity, why not just put it together and give her the highest chance? On the other hand, I haven't started Bev in my other 50% or whatever, and
then you got Prima.
Then I go Prima, or I could use
do
another
the-
strategy
yeah,
and start with PARP up front with chemo if I wanted to do that, and then and that would be the Velia trial strategy.
I don't think this is true, but I've heard some people say that PAOLA-1 is going to cause more Bev use. Do you think that's true? I don't think it is.
I don't know if it'll move the needle a whole lot on that. It might a little bit.
But people have their biases already.
I think the only thing it'll be is the high volume treaters is probably not going to change a lot because they've already looked at this data. The low volume treaters that haven't really thought about Bev being approved, because it was not the noisiest of approvals, owing largely to the fact that it was the third approval and it had been approved in Europe, so it kind of leaked out almost in the US.
And the biosimilars were coming.
Yeah, and there was a lot going on there. So I think in terms of promotion and so forth, so the people that are not as familiar with the literature, it brings to mind that, oh, you can use this up front, and a lot of experts are using it in certain patients. Maybe I should be thinking about what those patient characteristics are, and I should use it in my practice.
Yeah, so and then our theme, HRD positive. So in PAOLA, if you had an HRD positive patient, which now after you're done with the 6 cycles, you have that data, it increases from 16 to 28 months. So I kind of like that I start the Bev, and if she's BRCA or HRD, I lay her olaparib on, and if she's not HRD, she probably doesn't need it. Right. What do you think of that?
No, I mean, I think, again, looking at these medians, look at the numbers you're using. They're incredible, right? And that's what's so great about having all this new ammunition from ESMO.
Well, that's the only way we're going to justify the cost and the toxicity.
That's right.
If patients are really having long-term benefit, because we have to justify, again, the cost and the toxicity.
And it's still hard to really exactly pin down who these patients are versus the Prima population. You know, high-risk group in Prima, no doubt, based looking at the control arm. But then again, you're only up against placebo. So you know, how do you look at those number of stage 4s, so I think there's a lot to digest there. I really do.